Welcome to this video on the histology of the brainstem, brought to you by Visual MD. When examining a brainstem slide under a microscope, you'll need to identify the region, name key structures, and explain their function and position. We will cover the midbrain, pons, and medulla. The first section we'll look at is the midbrain, also known as the mesencephalon. A key landmark here is the cerebral aqueduct, which is a small, narrow canal connecting the third and fourth ventricles. A tiny circular lumen in the center is a major clue for identifying the midbrain. The midbrain has three main zones. The tectum, which is dorsal, is involved in visual and auditory reflexes. The tegmentum, which is central, contains motor pathways and nuclei. The crus cerebri, or cerebral peduncle, is ventral and houses descending motor fibers. The cerebral aqueduct is a narrow canal lined by simple cuboidal ependymal cells and surrounded by periaqueductal gray matter. Its function is to allow CSF flow between the ventricles. Clinically, obstruction can cause hydrocephalus. The periaqueductal gray, or PAD, is located around the cerebral aqueduct. Histologically, it contains dense neuronal cell bodies and is neuropil-rich. The PAD's function includes pain modulation and defensive behaviors. Look for neurons forming a ring around the aqueduct to identify it. Next is the red nucleus, located in the central tegmentum. It contains large multipolar neurons and appears reddish in fresh sections due to rich vascular supply. It functions in motor coordination via the rubrospinal tract. The clue is a large circular nucleus in the central midbrain. Here are additional histological images and a diagram showing the red nucleus. The substantia nigra is found between the tegmentum and crus cerebri. It contains dopaminergic neurons, whose cytoplasm holds melanin pigment, appearing as a dark band of pigmented neurons. It controls motor function via basal ganglia circuits. Degeneration leads to Parkinson's disease. It's the only place in the brainstem with dark pigmented neurons. The crus cerebri, or cerebral peduncles, are located in the ventral midbrain. They consist of dense, myelinated axons with no neuronal bodies. Their function is housing descending tracts like corticospinal, corticobulbar, and corticopontine. They appear as large, pale white matter bundles. Two cranial nerve nuclei are found in the midbrain. Cranial nerve 3, the oculomotor nerve, which controls eye movement, and cranial nerve 4, the trochlear nerve, which controls the superior oblique muscle. To summarize the midbrain, if you see a small central aqueduct, red nucleus, and substantia nigra, the region is the midbrain. Moving to the pons, the key landmark is the fourth ventricle, which appears as a large triangular cavity located dorsally. The pons is divided into two major regions. The basilar pons is ventral and contains pontine nuclei and fibers. The pontine tegmentum is dorsal and houses cranial nerve nuclei. The pontine nuclei are located in the basilar part of the pons. Histologically, they are clusters of multipolar neurons scattered among fibers, appearing as islands of gray neurons in white matter. They function to relay cortex signals to the cerebellum. Look for scattered neuronal islands to identify the pons. Transverse pontine fibers are bundles of myelinated axons that run horizontally. They connect the pons to the cerebellum via the middle cerebellar peduncle, appearing as striped horizontal fiber bundles. The fourth ventricle is a large triangular cavity lined by ependymal cells. The presence of a large open ventricle is a key clue, indicating the pons or medulla. 4 cranial nerve nuclei are found in the pons. CN5, trigeminal nerve, function, facial sensation, and muscles of mastication, chewing. Note, this is the largest cranial nerve and a massive landmark on the lateral pons. CN6, abducens nerve, function, lateral eye movement via the lateral rectus muscle. Note, its nucleus sits right under the facial colliculus on the floor of the fourth ventricle. CN7, facial nerve, function, muscles of facial expression, taste, anterior 2/3 of tongue, and salivary glands. Note, the fibers of CN7 uniquely loop around the CNVI nucleus before exiting. CN8, vestibulocochlear nerve, function, hearing, cochlear, and balance, vestibular. Note, located at the cerebellopontine angle, the junction between the pons and medulla. To quickly identify the pons, look for a large fourth ventricle, many pontine nuclei, and horizontal transverse fibers. The answer is the pons. Next, we examine the medulla oblongata. A key landmark is the pyramids, which are large ventral white matter columns. The pyramids are located in the ventral medulla and are dense bundles of corticospinal tract fibers. They function in voluntary motor control and appear as two large white matter bundles. Damage here can cause contralateral paralysis. The inferior olivary nucleus is in the lateral medulla. It is a large, folded gray matter nucleus that looks like a wrinkled bag. Its function is motor learning and coordination. Seeing this folded gray nucleus is a key clue for the medulla. The medial lemniscus is in the central medulla. This ascending sensory fiber tract is made of myelinated fibers and conveys touch, vibration, and proprioception. The medulla oblongata serves as the control center for the mouth and throat, housing cranial nerves 9 through 12. CN9, glossopharyngeal, and CN10, vagus, coordinate swallowing, the gag reflex, and taste. CN11, accessory, controls the muscles of the neck and shoulders. CN12, hypoglossal, manages all complex movements of the tongue. Together, these four nerves ensure seamless communication between the brain and the upper digestive and respiratory tracts. This summary table compares the key features used to identify the midbrain, pons, and medulla, allowing for quick identification of each region. Thank you for watching this video. Keep practicing those slides. Wishing you the best learning. I hope these visual landmarks make your next lab session much easier to navigate. You've got this, and I'll see you in the next video.